Hey everyone, so we are in week 5 of physiological psychology, and in the next couple of videos we are going to be talking about genetics and evolution. So let's start by just going over some reflection questions. You have most likely heard at this point in your academic career of nature versus nurture. So in other words, how much of any given trait or behavior is linked to our environment / upbringing versus how much of it is actually programmed at birth through genetics or heredity. And the book, our textbook really highlights that pretty much almost any behavior that has been tested has been linked to some kind of heritability. So one reflection question is, let's talk about TV watching habits. If someone is prone to watching a lot of TV, let's say 5 or more hours a day, how much of that behavior were they actually programmed with at birth or was inherited versus how much of it may have been attributed to their environment or upbringing? So just reflect on that for a moment. What would you think? So that particular behavior, television watching behavior, was one of the bullet points that the text highlighted that has been something that research has found evidence to support that there's an association between TV watching behavior and heritability. So again, the textbook highlights that most everything that's been tested has some form of association with heritability. But it's not quite as simple as that. The book also highlights that it's more complex. There's a balance between genetics, heritability, and our environment and upbringing. Let's start with Mendelian genetics. Mendelian genetics is actually called Mendelian genetics because a 19th century monk named Gregor Mendel demonstrated that inheritance occurs through genes. Up until that point, scientists had sort of thought that a sperm and an egg unite to create another unit, much like the way that when we mix two different colors of paint, we create a new color. They had sort of thought about it in the same way as that. But Mendel had highlighted that there's this concept of genes, which are units of heredity that generally come in pairs, one from our mother, one from our father. And they align along pairs of chromosomes. One exception to that is that male mammals have an unpaired XY chromosome. These are made up of DNA, deoxyribonucleic acid. I'm not going to ask you to say that or to know that particular word there, but it is important because DNA are basically our strands that serve as a template for the synthesis of RNA, which has a lot of different purposes in relation to our cells and what we do as an organism. And RNA is a shortened way of saying ribonucleic acid. So if you think back to some of our lectures on cells, we know the basic structure of a cell now. And within that cell, we know one of those basic structures are the nucleus, or what I call kind of the powerhouse of the cell, the metabolic center. And in that nucleus, we have genetic information, which we kind of had glazed over in some previous lectures, but this lecture is really going to talk about that genetic information. So we have chromosomes in the nucleus, and then we've got within those chromosomes the DNA, and a segment of the DNA is what we're talking about when we are referring to genes. So you may have already taken a human biology or general biology class and learned some of this information. So for some, this may be more of a refresher. But as human beings, we get 2 copies of every gene, one from our mother, one from our father. And the term allele references each variation of a gene that you may have or get from your mother or father. Alleles are often referenced as uppercase or lowercase letters. So in some examples in just a moment, I will highlight the different letters that are referencing alleles. If you're a visual learner, I put together this figure here to sort of help refresh our memory visually of what we're talking about here. So we get one gene from our father, one gene from our mother, or we can refer to that as an allele. If you think of this going back to the idea of the chromosome that's within the nucleus, and these little double helix here, symbols here kind of basically indicate DNA. And this little highlighted yellow portion here, that's what we're talking about when we say gene. And so again, one allele is from your father, one from your mother. So that's basically a pair of alleles that is what we're talking about when we say gene. So genes, our pairs of genes can either be heterozygous or homozygous. So heterozygous would mean that they're unmatched, and homozygous would mean that they're matched, or that they're exactly matched. So this is an example of what we're talking about here. So homozygous, let's say, and I told you I was going to highlight letters that might be referencing alleles. So let's say the capital B here and the lowercase b here are representing two different variations of any given gene. So an example of a homozygous match would be if someone had two capital B's, which again is one variation of any certain gene. And then, or another person may have two lowercase b's. Those are examples of the homozygous options for that particular gene. With the heterozygous option, that would be any given pair that is two different variations. So again, this be maybe the allele from the father and the mother, or here, this allele is from the father and the mother. So you can see there's different heterozygous matchings that can happen there too. And what we're really talking about here is any given trait that, we're talking about eye color, hair color, anything you might be able to observe, but also kind of going back to the beginning of the lecture, we're also talking about other things that maybe we can't see, maybe perhaps behaviors or habits that people might have as well. Another concept that you may remember from a previous class when we're talking about genes is the concept of dominant versus recessive genes. So dominant genes will have a strong effect in either homozygous or heterozygous pairs, whereas a recessive gene is going to only show effects in a homozygous pair. So normally when we're talking about the alleles, which again is the different variations of the genes, the capital letter is referencing a dominant allele versus the lowercase letter referencing a more recessive. As you can see, if lowercase is indicating a recessive allele or pairing, when they are matched together, that gene is going to be expressed. Whereas in a heterozygous pairing, the dominant allele is which gene is going to be expressed in that case. So in both of these cases, the uppercase B, which would be more than likely referring to the dominant allele, that gene, the dominant one, is going to be what is expressed in those pairings. So that's kind of what it's talking about there. So as was referenced earlier in this video, our biological sex that we are born with is different than how other genes pair up, because male mammals have a pair of an XY chromosome. So when we're talking about sex, we're talking about sex-linked genes, genes on sex chromosomes. And all the other genes that we have been talking about thus far we're talking about, they're also known as autosomal. Quick to get a quick sense. So when two humans procreate, a male and a female, a female is going to contribute an egg, and then a male is going to contribute a sperm. And though the egg and the sperm will unite to create the offspring. And so the male has the XY chromosome pair. And so when that, depending on what the male sperm is providing in that pair, will determine whether the offspring is male or female. So in this example, an XX would be female and an XY would be male. But it can be more complicated than that. There are times when sometimes genetically the biological sex of someone isn't quite clear because something happened genetically with the pairing. So just bear in mind that that is a simple way to think of it and gives you the basic concept, but that it can be more complicated and complex than what we're describing here. I also wanted to highlight that any biological given sex of someone is not the same as gender or the idea of gender. So sex, or the biological sex of someone, is referring to the attributes of a human or animal in terms of their biological pairing that they received when they were conceived. Gender refers to more socially constructed roles, behaviors, expressions, and the identities of any given person. And that can be very diverse. Some examples are that somebody born with a certain biological sex may identify as a different gender. And some people are more gender fluid and they don't really identify with one or the other more. They kind of identify with both. So those are just a couple examples. And I actually would like to post a video about all the different gender identifications that there are, which is different than the genetic biological sex that somebody may be born with. There's also this concept of sex-limited genes. So any given person, whether male or female, they will have a sex-limited gene, but if, let's say, for example, a male is more likely to express a certain gene than a female. And one example the textbook gives is hair on the chest. A biological male is more likely to have chest hair than a biological female, for example. The other example the textbook gave was breast size. Biological females are more likely to have larger breasts than a biological male. So those are a couple examples of sex-limited genes. So now I'd like to really expand on the concept of DNA. DNA is composed of two polynucleotide chains that form a double helix. So I kind of hinted at that earlier when I was pointing to those double helixes in the chromosome. And they carry genetic instructions for our development, functioning, growth, and reproduction. So it's pretty important, DNA. Just the basic structure of our DNA I wanted to highlight here. So we've got this sugar phosphate backbone, and then we've got base pairs. So that's a real basic look at DNA and just the basic structure. And now we're going to talk about these base pairs in a little more depth. So let's talk about these base pairs. DNA has 4 bases. So on this side here is our DNA because we've got that double helix. Okay, so we've got the 4 bases: adenine, guanine, cytosine, and thymine. Okay, so those are our 4 bases of DNA. Take a look here. And now take a look here, which we're going to talk about in just a moment. Take a look over here at the RNA. Do you see anything that's different? You may have already caught it that uracil is what is different here. So instead of thymine, it's got this base of uracil. So coming back to DNA, here's our 4 bases. And something that I think is kind of a neat way to remember which bases pair with which ones is apples in trees, cars in garage. So cars in garage help you remember that the C and the G typically go together, and then apples and trees would help you remember that adenine and thymine would go together. However, for RNA, it would be a little different. You'd have to think of something different for A and U because U is obviously different than T. So coming back to DNA, the order of the bases and how they're paired determines the order of the corresponding bases along the RNA molecule. So that's where we come into the RNA molecule here. So what is happening here is basically going to determine what happens over here. The order of bases along the RNA are important because they are going to determine the order of amino acids that compose a protein. So one example the textbook gives is that if 3 RNA bases are perhaps in the order of cytosine, adenine, and then guanine, then a protein will add the amino acid glutamine. And so it basically determines what amino acids are being created, which will ultimately determine what's going to happen. So more on RNA, ribonucleic acid, which again, I'm not going to ask you to know that big word, but I just wanted to be sure to mention it. So some would argue that RNA may even be more important than DNA, because even though DNA is doing all of these important things, RNA is really what is needed to carry the messages from the DNA, because DNA generally has to stay in the nucleus of the cell. So RNA, which one example that I give here is messenger RNA, will carry messages to ribosomes. So without that RNA, what the DNA is telling the cell to happen, the ribosomes wouldn't receive that message, for example. So RNA is essential in various biological roles: coding, decoding, regulation, and expression of genes. And I did find a pretty neat video that really goes into the different kinds of RNA that I found very interesting. So I'm going to post that. Be sure to check that out. But this figure here is from our text. And it sort of gives you a quick visual on what's going on when we're talking about DNA and RNA and how it relates to development of an organism. So here's our DNA. You see that double helix. And we've already talked about that it's determining the pairings that are happening here determine the base of the RNA. So here's the RNA, a copy of one strand of the DNA. So it's been created based on the DNA. And based on that, the order in which the RNA has been created is going to determine which amino acids create which kinds of proteins. And so here we've got a triplet of bases determines one amino acid. So here's the triplet of bases determining one amino acid, which ultimately is going to create a protein. And some proteins become part of the actual body structure, and others are enzymes that control the rate of chemical reactions. So it truly is controlling the development of an organism and the different things that we're doing. Let's talk about mutations. So is DNA and RNA always just a really clear-cut thing that's going on, and we know, okay, these different patterns are going, and that's just how it's going to happen? No, absolutely not. There's going to be sometimes things that don't quite go the way that we thought it should go. So mutations are heritable change in the DNA molecule. So it's actually something physically that has changed within the molecule itself. So the main examples that the text gave were duplication and deletion. There are other types of mutations such as inversion and translocation, Inversion, just briefly, is when there is a segment of a chromosome that is reversed, and translocation is where segments of a chromosome may break off and join a different location. So the textbook doesn't really get into those, so it's just kind of for your information. The ones that the textbook really highlight are the duplication and the deletion. So here's some examples for anybody who's a visual learner. Here's an example of deletion. So as you can see here, this B is missing over here. So it's an example of a deleted piece. And the duplication would be, for instance, here, 2 B's here. So the textbook also explains that human beings have been around for a while. So any mutation that happens is most likely not going to be advantageous. That's not to say that a mutation couldn't be advantageous. And so it's just kind of highlighting that more than likely it's not going to be advantageous, but it could be. One example how that relates to psychology is schizophrenia and chromosome 22. So some different research studies have indicated that chromosome 22, when there is a microdeletion that has happened in that chromosome, it has been associated with a small percent of people who suffer from schizophrenia. So now let's talk about epigenetics, which is the study of heritable changes in gene expression. So when we think about genes, the activity of the different genes that we are born with can vary. Some genes are essential to our development. So as a fetus, they may be super important, but as we get older, they become less important or maybe even become inactive. And some may be very active in one person, but not so much in another person. So gene expression can vary from person to person as well. Experiences that we have as human beings can turn a gene expression on or off. And that expression can actually be altered by experiences. And those differences that may happen within us due to an experience can actually be inherited by offspring. So that's what we're talking about with epigenetic changes that can be inherited. So one example that the textbook gives to kind of bring that to life is a study that was done with mice that were conditioned to fear a certain smell. So researchers found that these mice who had been conditioned to fear a certain odor, their offspring were actually more sensitive to that odor than mice that hadn't been conditioned to, whose parents hadn't been conditioned to fear this odor. So that's one example that kind of highlights how epigenetic changes can be inherited. I'm wondering, how can one's experience actually change how a gene is expressed? How can an inactive gene expression become active by an experience? So the answer in a nutshell is that it's a chemical alteration. So this change in the chemical environment of the brain and the body is going to impact gene expression. So one example that the textbook gives is that acetyl groups may be added or removed, which will in turn loosen or tighten the grip of histones on DNA. And now you may be wondering, what does she mean by histones? This is the first we've talked about it, and that's true. So histones are proteins that bind to DNA. And when we were looking earlier at DNA in general, we were looking at these figures that made it look like a straight line, when in reality, DNA is shaped more like a string wound around a ball because of these histones that sort of have this bind on it, or this grip on it that shape it that way. So when those histones either loosen or tighten their grip on the DNA, that's going to facilitate a difference in how that gene is expressed. So let's take a look at these histones. So this is actually from our textbook. It's figure 4.5. So we've got this tail here, and then as you can see, it's more like looks more like a ball of yarn, or like I was saying earlier, almost like a string wrapped around a ball. So we've got the DNA, here's the histones. And so if we go back to that example of a chemical environment change, if an acetyl group, for example, is removed, then that histone is actually going to tighten the grip even more. And if the opposite occurs, like for instance, an acetyl group is added, that histone is actually going to loosen the grip. And so the gene expression is going to change. So if you're a visual learner, I just kind of wanted to show what these histones look like. I wanted to end on this figure for our lecture on DNA and genetics in general, because I like how this figure sort of ties a lot of the different concepts we've talked about together, and I wanted to be sure you guys had access to it. So this figure sort of ties everything together by showing us, again, we've got this chromosome, which came from which structure of a cell body? The nucleus. Hopefully you said the nucleus. So we've got our chromosome here, and as you can see, we've got these strands of DNA, our double helix, and there's our histone that we just talked about that's kind of winding that DNA into a string around a ball, so to speak. And then we've got it even breaks it down further here into our pairs, which if you remember, we've got apples and trees together, the adenine and the thymine, and then our cars in our garage with the cytosine and the guanine. So those are our actual bases of the DNA pairs. And then it breaks it down even further to show us that phosphate sugar base here.